Okay. And I'm allowed to use the mouse or destroy the no, it's fine. Yeah. So hi everyone. Thanks a lot for the invitation. It's really a great pleasure to have this opportunity to present my lab work. And so I'm a researcher at LISN in Paris-Saclay University. And so today I will talk about two the main focus of my lab. The first one is inference of evolutionary history. So it's how from observed genetic variation of present-day individuals you can retrieve past evolutionary forces such as demography or selection. And the second topic will be a description of contemporary genetic variation and how you can learn from large datasets. Okay, so in the first part, I will basically talk about supervised learning for demographic inference, and I will mention approximate Bayesian computation and deep neural networks. And I will focus on the design of such networks. And in the second part, I will talk mostly about unsupervised learning and the use of generative models to create genome sequences. Okay, so demography is a lot of things. It can be migration between population when population diverge, our population moves in space, and so on and so forth. And here I'm really looking at a very specific subtask of demographic inference, which is reconstructing effective population size through time. So if you look at here on the left, it will be a case where a single population had a very high effective population size in the past, and then this declined drastically, then the population size was more or less stable and then expanded again in the very recent past. Okay, but that's not what you observe, it's completely unknown, and what you observe instead are sequences for multiple individuals. And here are all aligned, and you have millions of markers along the genome. And you want to reconstruct the population size history, which we simplified as this stepwise constant function of population size. And because in population genetics, we very, very rarely have ground truth, so we don't have label data. The field is using a lot model-based inference. So the model would be, for example, an evolutionary model. And here in particular, we do model-based inference, but we even do simulation-based inference. So you can use your model to simulate data according to demographic histories that you would have specified, and you end up with many data sets that are this time labeled that you can use to train a model that will predict demographic history. Okay, and so what has been done a lot for the past 20 years, maybe in population genetics, is to summarize the data set, which is very large, into a small subset of summary statistics, and to do inference using these summaries of the data. Okay, and because you might not all be familiar with population genetics, for example, linkage disequilibrium correspond to the correlation between SNPs along the genome. Side frequencies spectrum correspond to the allele frequency of mutation in the genome. Identity by state will be looking at how much of the genome is shared between two individuals and how big are the identity tracks and so on. And so all these statistics can be used in an approximate Bayesian computation method, which basically compares simulated data, so the dots, to the real data, the stars, but compare them in terms of summary statistics. From that, you can keep only the simulations that are close enough, and that allows you to draw a posterior for the demographic parameter in this case. Okay? The issue with that is that you need to specify a set of statistics, but you also need to define a distance measure, a tolerance rate on how many simulations you keep. If you don't keep enough summary statistics, then you will lose information that is in your original data set. If you use too many summary statistics, then you're in the curse of dimensionality world, which means that it's going to be hard to simulate data that are close enough to the real one. Okay, so because of that, people have started using more and more summary statistics so as not to lose information. And instead of using this classical ABC with a rejection step, they used a deep neural network, which didn't have this issue with the curse of dimensionality and picking the distance. And the goal of the neural network is, as before, to predict here demographic parameters, which at the end you can add or not an ABC step that would allow you to get the posterior. Okay. One example, which if you've seen Janet Kelso talk on Keynote on Sunday, you know that people are very interested in inferring admixture between archaics and modern humans population. And so this paper used a deep neural network based on summary statistics to investigate whether there was a third archaic introgression in Oceania. Okay, but as you've seen, it's still a bit biased because you need to know the summary statistics. So this in machine learning would be called like expert features or encrypted features. And so for this reason, the question was, could you use a neural network directly on the original data set to compute for you automatically the features? Okay. So the goal still is the same, inferring parameters, and you can still add an ABC step afterwards to get the posterior. Okay. And so, it's quite recent. I think the first paper was in maybe 2018 or so that really tried to use directly the SNP matrix. So because I'm in the population genetic world, I simplify things a bit. So I'm just interested in position that where at least one individual in my collection differs from the other, and also simple position where there is just like one nucleotide that change. So that's SNPs, and so for this reason we can represent the data as this black and white matrix here. Okay, and so people have been using here, for example, convolution. And calling that on the left the SNP image matrix. Okay. This is one work where they use that to infer introgression, but also recombination rate along the genome, I think selection, many things. And here are some reviews that you can look at if you want to know a bit more about population genetics application of the neural network. But now I will talk about what we've been doing in my lab, which is comparing previous neural network and one that a few that we designed. So comparing them together and also comparing them to the state-of-the-art ABC approach using summary statistics that is, very good already at inferring population size, and also see how we could combine all of that. And so first thing that I want to highlight in why we designed the network that I will show is that I said people were calling that the SNP image, but that's a bit misleading because contrary to a regular image where the kind of the vertical orientation has a meaning, for us individuals can be permuted and we should get the same answer, because they're just like a collection, a pool of individuals. So if I permute the line of this matrix, I should actually get the exact same answer because they correspond to the same population. Okay. If I permute the columns, then it's not the same because we know there is some dependency on the genome that makes sense, and that is actually very informative. So there are different ways of being invariant, and people have proposed, for example, to enforce a ranking on the lines that you have to specify and that you apply to any matrix. And so in practice, it actually works well, but we know that in high dimension, it can be hard to find and design a ranking that is very stable. Then, of course, you could just shuffle, so do what we call data augmentation, like people rotate images. In this case, would be shuffling lines and let the network learn that, in fact, the line order does not matter. But what's a bit sad about that is you will use, computational power to learn something that you know very well. So the third solution, and the one we took, is to encode the invariance in the network, so that by design your network will be invariant. Okay, and so there is a paper that's called DeepSet that shows that if you stack, layers that are equivariant, meaning if I permute the input, I permute the output the same way, and at the end I stack some invariant function, then provided that I have enough layers and so on, I should be able to approximate any invariant function. And this has been used by this paper that estimates recombination rate along the genome. And this gray invariant operation was a mean. And what we've been doing is that based on Lucas' paper that showed that you could, instead of waiting, for the very last layer to put the invariant, you could do that at each layer of your network. So that's what we did. So now each block computes equivariant features, but also invariant feature, and that allows to basically compare the individual information to a global context. And this is still, the equivalent function is still just a simple mean. Okay, and we also use this global invariant function to do prediction through the network. And so I will not comment on all the results, which are like many comparisons of many networks. I just want to comment on a few things, which is found that the ABC, just summary statistics, is performing very well. Like networks that are very stupid design are performing very poorly, which is reassuring. And the importance of designing properly your network. That you could almost reach, you reach similar performance than ABC without using any encrypted summary statistics. So you could bypass, in a sense, the summary statistics, but still not as good as the ABC and summary statistics. And that if you combine features computed by the network and encrypted feature, you're a tiny bit better, but it's like a slight increase in performance. But which could mean that there is some information missed by the summary statistics, but we have maybe a bit of a hard time to get it with this network. And we applied that to some simulation where we have a constant population size, so the black lines are the truth, and the blue envelope is the posterior distribution that we get by applying the ABC on the network. And you can see that you can retrieve like constant population size, decline, expansion, and some more complicated scenarios, but not always extremely precisely. So if you have like very sharp changes, it might be hard to predict exactly these changes. And the good thing is that the posterior, you can check if it's well calibrated, and it is. So meaning that if you give, say, 90% credible interval, it is actually, you have actually 90% of the chance to have the true value in the interval, which is what you want. And so we apply that to cattle population, and we found that the history is kind of stable with a slight decline that could correspond, I mean, so it's correlation that could correspond to the time of domestication. Okay, so the question was, are we missing something there? Like, is there more information that we did not manage to get with the network, or is there actually not more information in the data? And so we thought that this average equivalent function was a bit rough and maybe a bottleneck in the information flow. And so we decided to replace it by something that would be more expressive. So this is an attention hub, so we call it attention hub, and it's really related to agglomerative attention for those who know that. And basically the idea is that instead of having all the individuals contributing exactly the same way, in fact, we want them to contribute based on what's their genetic sequence, and that you can do through attention. So you have hubs that will be this invariant function that before were just the mean, okay? And each individual can vote for hubs based on its genetic sequence, which is basically a fancy way of saying that you're going to do a weighted average, but based on the sequence and not based on the place of the individual in the matrix. So now you're going to have this function that is still, permutation invariant. You can move individuals in the matrix and you still get the exact same answer. And then you can later distribute, the result of this invariant operation back to the sequence, again through an attention mechanism, so that at the end you have here the equivariant features. Okay, so that allows you to learn richer statistics than a simple mean. For example, it can focus on a subset of individuals, and from the hub you can, like before, predict directly the global population parameters, such as the effective population size. This is not quadratic in the number of individuals because you have the hubs in the middle, and it's also adaptive to the sampling size. So if you have different sampling size for different population, that's very convenient because you don't need to retrain a new network. And briefly, the way it works is that for individuals, you can compute keys and values that are just, you do that through a trainable mapping from the sequence to this key and value, for example, with convolution filter. And then the hub is asking for specific patterns through the query, and the query is also a vector of trainable parameters. Okay, so applying that on the same setup as before, you can see that by using this and some small variation that I don't have time to explain, you have a gain of 40% compared to the previous deep neural network. So this is just deep neural network only on the original data without similar statistic and anything. So clearly it seemed that there was a bottleneck in the way the previous architecture was designed, and also all the other CNN and networks from the literature that we tested. And so we applied that to the human genome diversity panel that has population spread a bit everywhere in the world. And that's a good use case as well, because it has, whereas talking about this property that population might have a different number of samples. And so, for example, here, result on the central, so South Asian population, you can see that far away in the past, you share the same history, which makes sense because all the population were actually merged into this ancestral population. And then more recently, there seems to be an expansion in many of the population that could be linked to this Neolithic expansion. And so we spent some time in the lab developing a tool that we hope can help the community to benchmark and share more easily networks, because there's many papers out there and it's not always easy to, sometimes you have to recon the network so that it's really applied to your task or to your data. So the goal of this software is really to kind of push people to have a more reproducible work, and they can share either pre-trained or not trained network, and they can also use the tool to design new networks, but in a controlled environment. So to summarize this part, I showed that the way you design the architecture matters, and you can think about architecture that are tailored for generating data. You can use them to reconstruct effective population size without relying on handcrafted features. That you can combine that with ABC to get a posterior distribution. And training of neural network is always a bit intensive, but as it's applicable immediately to multiple population, which is not the case of most ABC technique, you will then have a huge gain in computation time, and that will develop the software to help sharing all of that. So the second part is a bit different philosophically, and so here we're interested in the genetic variation that exists in individuals, and that you can observe in data that you have access to, but also in data that you might not have access to. And I want to highlight really that it's a project that is done, that has been designed from the beginning with Burak Yelman. And so just first note, we wrote a review on the use of generative models in functional and evolutionary genomics, which focus on different types of generative networks and applications such as dimension reduction, prediction, or generation. So here is one of the examples from the paper where we comment on this BATE paper that uses a variational autoencoder to reduce the dimension of the genetic sequences to a latent space, a small latent space that they can then visualize, and here they show the structure of that's human population that kind of seems to correlate with geographic location on the map. And so what we've been doing in the lab is to work on generative adversarial network, BAE, and restricted Boltzmann machine to do generation of genomic data. And why did we want to do that in the first place? Like, where did the idea come from? Is that there are more and more data sets, but in fact they are not all easily accessible. Some are completely private, some are very hard to get access to, and so there is clearly a lot of information, and in particular it's been studied that some populations are highly underrepresented in the public data sets. So we were wondering if we could augment the public data set by using synthetic artificial genomes and kind of lower the issue with releasing sensitive data. And so the idea was to use a generative neural network to generate samples that would have the same distribution as this private data set. And the first question is, okay, do these generated samples retain enough the characteristics that matter in population genetics and that we could use for example, to do selection, detection, imputation, ancestry inference, and so on. And so the first the first model we used is GAN, which basically wants to generate samples from a distribution without explicitly defining it. Okay, and so what they propose, so that's Goodfellow and colleague, is to sample from a very simple distribution and to just learn the mapping to this a more complex real distribution. And the way they learn that is with a neural network that is called a generator. So that basically goes from noise to a good-looking generated sample. Okay, but how do you learn the parameter of this generator? Here, you need to use a second network that is a discriminator, and that takes as input either fake or real data, and learns to distinguish between them. Okay, so you can see that basically it's a game between two players that have opposite interests. So the generator wants to fool the discriminator, and the discriminator wants to beat the generator. And in this way, you can optimize all the weights of the networks. So we trained such a fully connected GAN on 1,000 genome data, again, human population spread worldwide. And once it was trained, we could use the generator to sample, to generate new pieces of genomes. And what I want to highlight is that these genomes have a one-to-one correspondence in terms of SNPs with the real data. So they can really be merged together. Like the first SNP of the artificial genome corresponds to the first SNP here of the real genome. Okay, so if this part of the real genome is public, and this mimic a private data set, you can augment the public database. Okay, and so if you, for example, look at the PCA between using artificial genome and real genome from the 1,000 genome, you can see that some models are very poor at retrieving the population structure, and some models are way better. And so the second network is a restricted Boltzmann machine that is quite different philosophically, because it's again a network, but so it's a two-layer network with the visible layer where the genetic data lie, and a hidden layer that is basically a latent space. And there are interaction only between the visible and the hidden nodes. And basically there is a probabilistic model that is depends on an energy function. And this energy function has weights which are the weights you're gonna optimize during training. And so the goal is from a flat energy function, to go to this energy function that is low for good genetic sequences and high for unrealistic genetic sequences. So on digit images, it would look like that. At the beginning, you can so you can use the RBM to generate samples, and at the beginning it will just generate noise, and it will slowly go to generate good-looking digits. Okay. So we, in the first one that's been published already, we evaluated the quality of the generated sequences by using population genetic classical summary statistics. So I talked about them before, the linkage disequilibrium, which is just the correlation between two SNPs along the genome, allele frequencies, more complex haplotype structure, and so on. And we found that most characteristics were quite well preserved. We had, for example, some issue with very rare alleles. Not everything was perfect, but overall quite good. And something nice that we could do with the RBM is that you can visualize what's in the latent space, and you can also look at what activates the different hidden nodes. And so, for example, here you can see that some nodes are activated by all population except for some parts of the world, and this one are activated by all African population, to a lesser extent by Afro-American, ethnic population between European and African here, and not at all by the other population. Okay, and so one proof of concept of an application that you could do with this synthetic genome is that if you want to impute a sequence, so say here a sequence of an SNN individual, Ideally, you would want to have the 1,000 genome, that is a large database, and also the Estonian dataset, but this one is private. So in practice, you will have just the public dataset. But what we suggest is to augment the public dataset with GAN or RBM synthetic data. And so if you look here, that's the how good you are. And so that's a classical platinum imputation that it's always quite easy for common alleles, but for rare alleles, it's not that easy. And so what you can see is that it's always better to have, of course, the full Estonian private dataset. But if you have the artificial genome on top of the 1,000 genome, then you will improve the imputation. So, okay, that's nice, but of course, it would not be fun if you don't have some issues that you need to fix further. And so one of the issues is that since it was a fully connected architecture, it went well for, say, simulating 10,000 SNP that cover 2 million base pair of the genome. But if you want to scale up, that's going to be a complication, right? So you're going to increase drastically the number of parameters, and that's not feasible. So in a recent preprint, we proposed two novel architecture. One is a conversion on GAN, which I will detail a bit more after. And this new architecture allows for example to go to only 7 million of parameters instead of the 238 million we had already for just 10,000 SNP. And then the conditional RBM, which will which will generate a chunk conditionally on the previous chunk of the genome. And that's more for technicality, but just to say we also changed the GAN to the classical GAN to a Wasserstein GAN because it's been shown to be way more stable in terms of training. And so that's sort of a schematic view of the GAN, and I just want to pinpoint maybe three parts. That is okay. That's classical where you have an increase of size with deconvolution and a decrease of size with convolution. Okay, the latent space is in red, and so you can see that it's not only at the beginning, but it's like at different layer, and that allows to have a more hierarchical learning of the latent space representation. But importantly, you have this blue here vectors that are called location-specific vectors, and this idea came from an OSMI paper, and they did that for an autoencoder. And that's very important because, okay, the convolution makes sense for our genetic data, but in our case, and they allow to reduce the number of parameters, but in our case, we don't want to be completely shift invariant because then we would forget what SNP we're at. And as I said, like it's very important to understand what's the SNP because it corresponds to a real SNP in the real world. So you have to keep this. Okay, I see what's no. Okay. Is it telling me that I'm late? Okay, so you have to keep this to have this real SNP correspondence. The conditional RBM, very briefly, it's on the left. Okay, I don't have any pattern in mind. On the left, you have a classical RBM, and on the right, you have an RBM that is trained in a way that it's always conditioned on the first half to predict the second half. So you can see that in a sense a bit like using RBM for imputation, but it's trained this way. And then at generation time, you can generate sequentially by first generating the first chunk, then taking the two red values for the second chunk and conditionally generating the second part of the second chunk, and so on and so forth. Okay, and so here are some results on the large data set, so which is 65,000 SNP that cover, I think, something like 80 million of base pair. And so you can see that again we're able to retrieve the population structure the allele frequencies, and in fact, we improved. I told you that we had some issue with very rare variant, and this is not the case anymore. And here is the ID, and you can see that there is still some room for improvement because at short distances we don't retrieve as much correlation as we should. Okay. And I don't have time to present you all the details, but so we compared previous and new model on the 10,000 SNP because that's still doable. They were not scalable from the 65K SNP. In terms of many summary statistics, and also in terms of overfitting and underfitting, using a score that is very interesting from this paper if you want to look at, and that is based on distances between samples, and basically you want the generated and real distribution to be mingled correctly, and also perform some black box membership inference at that. And so we saw that there was maybe a small leak of information for the RBM that has slight tendency to overfit, and it's hard to prevent him to do so. While the GAN is a bit underfitting, although on the summary statistic, they still look quite fine and useful for population genetics. So that's to summarize the second part. So I showed that we could use GAN RBM. I didn't show BAE, but we also tested those to generate data and try to augment public data sets that we could scale up using novel architecture. And in fact, the novel architecture also improved the produced summary statistic compared to the previous one. And finally, that the application could be in visualization, so in the latent phase, imputation, selection scan. I didn't show, but in fact, you can check that you still have the selection signals in the generated genomes, encoding, of course, and GWAS, it's more like a future work still has to be proven. And very importantly, you need to check, because it's easy to generate things, but you need to check their quality, and you also need to check that you're not, overfitting, otherwise the whole purpose is not matched. So, if you want to see more work on generative models in genetic, you can look at this review for inference at the 3+1. That's all the literature I said from the lab. And really, I want to thank all the people who actually did the good part of the work, in particular Flora Jay, Théophile Sanchez, and Pierre Jobic. And yeah, thanks to you all. And if you have any questions, I'm happy to answer. Okay.
Yes. Thank you. Really interesting talk. I'm wondering, earlier today, Carl Müller was mentioning that if you train an autoencoder and you feed it its own output, it tends to converge towards actually a training or a point in your training data. And I'm wondering if it's different with variational autoencoder, because then if you train such a model to kind of make artificial data, but there is encoded somehow in your model, you didn't realize that earlier, then you could go back to the prior data set and actually compromise safety of the data, right?
Yeah, very good point. So we didn't try exactly that, but so we tried what I mentioned at the very end, which is the membership inference attack, where you train a new GAN, in this case, on the generated data, so it's a bit similar in spirit. So you train the new GAN on generated data, and then you use the critique of the GAN to score samples. And basically, if you leak information, then the samples from the training set will have a different score than the sample from the test set. And so that's called membership inference attack, where you can distinguish between training examples and test example, which is a bad thing. Although it means that still the attacker needs to have the sequence in the first place, So it's kind of a nice setup for the adversary, but yeah. And so we saw that for the GAN, in fact, there was very little leak, and it was very hard to distinguish between. So the accuracy was around 50%. While for the RBM, and that's why I was saying it was slightly overfitting, probably, there is a leak. So if you take like the top 1%, then they will be very often a training example.
Thanks for the interesting talk. My name is Mohammed Al-Buda. So I like the idea of generative genomics, but actually it is the same danger as any AI thing. I think the big genome project may use these tools in future to put in the database the generated genomes, not the real ones. And at the same time, they will preserve the genome structure. So this can be really interesting things that can happen in the future. I see this happening in this genetic recognition of genetic disorders based on facial features. And now the company is now, instead of putting real patient faces, they put generated patient faces. I think maybe we'll go in this direction in future, and hopefully this not happens. Yeah.
So your question is like, you mean would
No,
I?
I see this might be very interesting in future that people may submit in database generated genomes on the real ones. I mean,
Yeah.
yeah, this would be very interesting. So my second question is about the imputation, because you mentioned that you can generate genomes that can keep the population structure, but actually, I think the dilemma still exists because populations that are not well presented, they don't have enough data to generate. For example, we deal with population data from Africa and Middle East, and they are less represented. And to do imputation, we still use the 1,000 genome, which is not correct. Again, actually, it is like that we mapping our genome back to the other population and then use the same result. I think the only way to solve this is to have more sequencing activities, to have more genome sequences from other populations. So I think still the problem is difficult, but I think it's a practicality if your tool can help to take, for example, exome data and impute it in a very more efficient way to genome data for the same population, provided that the number of whole genomes is much less than the number of whole exomes. For example, because of the cost issues, many populations have large number of exomes, few number of genomes, and if your tool can use this information to help impute the exomic data, this may be solve the population-specific problems.
Yeah, very interesting. For the second point, I fully agree with you that in fact there is really a need of push towards collecting, more ethical way of collecting the data and thinking about diversity, and this will not replace, doing this effort, because if you just have like two genome of a population, that clearly won't work, that won't be good at all. So I agree, but it's that there are some private data sets that do have a few of these samples, like 23andMe and all these companies. They do have quite a lot of individuals. In fact, they just don't share it. Yeah.
Okay. One last question, because Flora Jay, please.
Hi, thank you very much for your talk. I was wondering, since you mentioned the part of these artificial genomes can be used for the potential research or imputation. Have you tested these artificial genomes for applications in imputation for low-pass coverage whole genome sequencing?
For what?
Low-pass coverage.
So I mean, so that's here. Where was it? Here what I'm doing is exactly that, which is I assume that I have some data with low coverage and lots of missing information, and I impute these sequences using the artificial genome. Is that what you're asking?
Yes, it's just that currently The current approach is by using genotyping arrays, but now that we are reaching to the point that genotyping arrays are going to be obsolete, one alternative is by using low-pass coverage genome sequencing for application, especially in studies. But the problem with using sequencing data is that most of the methods for imputation do not work because sequencing data has different properties. One of them is the coverage, because the more coverage you have, the more confidence you have in the SNP. So.
And maybe we can discuss more, but I mean, this was done using like Impute2, which is one of the classical tools for imputation, and that can be applied to sequences. But let's talk more because there might be some subtleties that I don't get.